right. Um, this is Anna, and we're going to be continuing on with our neurophysiology lectures. And we're going to be working on part 5, where we're going to start getting into the physics of how electrophysiology works with neurons. All right, so let's just kind of start with a blank slate and kind of talk about what we're doing. So, like, the big questions that we're going to be asking is, how do we generate an electrical signal? Okay, so if you remember, we've talked about this before with lightning or power lines. You're looking at free electrons, okay? But in animals and plants, free electrons damage DNA, okay? So you don't want to use that. So instead, we're going to use charged ions. All right, some examples are sodium, calcium, potassium, chloride ions. All of those have a charge to it, okay? And then the next thing we're going to be looking at is, how do we transmit from one structure to the next? Now, we've already looked at this with the muscular system, where we looked at the neuromuscular junction. It's essentially the same process, but now we're going to be dealing with two neurons, or a neuron and an astrocyte, etc. Okay? So let's talk about some of these definitions that we have. We have this idea of a human charge. Okay, so we have charges within the body. If you add them all together, the human body is essentially neutral. Okay, but when you concentrate charged ions on either side of a plasma membrane, then you can measure voltage changes, okay, because that concentration on either side of the membrane is the biggest component, okay? And so we're going to be using this energy. So voltage, what is that? Another way of thinking of voltage is the potential for energy to happen, okay? So if you remember, I did this with the muscular system, where, I drew a cell, okay? So this is your plasma membrane right here. And I'm going to put a bunch of ions here, and then I'm going to put ions here, but not as many, okay? Then I can have a voltmeter, and it can take one little electrode and put it right here, and take another little electrode and put it right there, and it will measure the difference between the two locations. Okay? And what it'll do is tell you how much charge there is. Now, if you're dealing with a car battery, you're going to look at it in terms of volts. But in humans, we're not that energetic, so we're going to do it in millivolts instead. Okay? All right. Current. What is current? Okay? Current is describing the flow of charged ions from one spot to the next. Okay? The higher the voltage, the higher the current, because it still follows, it's going to follow its concentration gradient. Okay? From high charge to low charge. Now, if the voltage is the same between 2 points, exactly the same number, then no current flows, because it's the same. Okay, so you need to have that differential between the two for it to happen. Then you've got the idea of resistance. Okay, so this opposes the flow of ions. Okay, if resistance is high, so if R is high, then current is low, okay, and vice versa, okay? Excuse me, no, not vice versa. If resistance is high, then current is low. If resistance is low, then current is faster, okay? It can flow easier, okay? Now, plasma membranes, so I'm abbreviating PM for plasma membranes, somewhat resistant because they're made out of phospholipids, and oils and fats and waxes are resistant to charged ions. But it has lots of transmembrane proteins embedded in the plasma membrane, okay? And these proteins, whether they are a leak channel and passive, or whether they are a gated channel, these all let charged ions flow, and this will decrease the resistance, okay? Now, you may have heard of the concept of an insulator versus a conductor, all right? And we talked about this in elementary and middle school and high school science. Insulators have high resistance. Conductors have low resistance. And what that means is stops energy from leaking out and helps energy flow faster. All right. Now, with electrical lines, things that are rubber, plastic, glass, these are all insulators, okay? Our phospholipids can act as an insulator. So in our body, phospholipids can act as an insulator, which is why with the myelin sheath, we use multiple layers in order to prevent signal from leaking out. Now, in the world, we use like copper wires to help with conduction, all right? In the human body, axons and plasma membranes can also be conductors, okay? So for this to act as an insulator, you have to have lots of layers, okay? For this to act as a conductor, you need channels that let ions through. Okay? All right, let's go ahead and look at a picture.
All right, looking at this picture here, what we're looking at is the voltage measured between 2 points. So what I like about this particular picture is the way they're showing the different ions, okay? So let's kind of blow this up, okay? So you've got this really nice plasma membrane here, okay? And then they're showing positive here, and they're showing negative along here. They're doing green for negative chloride ions, okay? They're doing, this is your sodium, which is a positively charged ion, and these are your potassiums, which are positively charged ions. Now, what you've got is a higher ratio of positive ions on the outside. Okay, so even though I've got chloride ions here, notice that they're not right up against this area. So they are not playing a major role in the voltage right here. What's playing a major role are the sodium ions versus the potassium ions. And I've made a mess, so I'm going to kind of erase all of that so you can see it better. Okay? Now, this creates the polarity that you need to maintain the negative 70 millivolts. Now, you remember with the muscular system, we were using negative 90 as the standard resting membrane potential. Okay, but here we're going to use -70 with this particular neuron. Okay, now the other thing I like is the way they have embedded channels. So I've got a chemical gate for potassium. Okay, I have a chemical gate for chloride ions. I have a chemical gate for just general cations. Here's another potassium gate. Now, these chemical gates are responding to neurotransmitters. The neurotransmitters open them, they come in, and everything is fun, okay? I guess this one is responding for sodium primarily. That's what they're using that one for. Okay. What you will notice is for all of these, this one, this one, this one, this one, and this one, all of them are chemical, which means they do not respond to voltage. All right, so this is the receptive segment of your neuron, okay? But when we get down to this area, and they're not showing them here yet, but there will be additional voltage gates all in here, okay? And these are going to be voltage gates. And they will have voltage gates. So this one they're showing that is a voltage gate for sodium. And then up here is a voltage gate for potassium. Okay, so I'm going to erase this so that you can kind of see what we're doing here better. Okay, so you got to pay attention to those gates. So this is the section that can send a signal. Okay, because it has voltage gates, which will allow a flood of sodium in, and for repolarizing, the potassium will go out. Okay. All right. I think we can look at the next slide now. Oh, actually, let's move down here. So the action potential, when it is propagated, so that means if you've propagated an action potential, it means these are opening automatically because of a voltage change. And a voltage change is another way of saying depolarization. And in this case, it's going to be an action potential. Okay? And that's going to travel down here and travel down here. Now, I've got another gate right here, and this is a voltage gate for calcium. When I depolarize here, in response to this action potential that's going down, these calcium ions will go into here. The calcium ions will then trigger the neurotransmitter to leave the cell. Okay, and this is going to be the signal that is secretory. All right, let's look at the next slide now. All right, let's quickly review gradients. Okay, so gradients go from high to low. Okay, and we can use different types of gradients. They can be concentration gradients, they can be chemical gradients. So let's talk about those a little bit. Okay, so you've got concentration gradients, which are chemical. Okay, ions are going to diffuse passively from high to low. You have concentration gradients that we call electrical, but by electrical, we're actually talking about charged ions, okay? Which brings us to this idea of electrochemical, okay? Electrochemical are concentration gradients, and remember, charged ions are technically a solute, so they are, well, not really a chemical, but a chemical. So you're basically going from a high ratio of charged, and here's the key word, charged ions, to low. And things are going to follow this concentration gradient as long as a gate is open. All right, let's look at the next. I think we've got pictures next. Okay. All right. When we are looking at this electrochemical gradient, it's going to be determined by the ratio of these charged ions in the different areas. So if we look at this picture right here, they're showing this whole area as the interstitial fluid. So this is outside the cell. If we look at this area, it is inside the cell. So this is the cytosol. Your cells are designed to keep a higher concentration of more potassium on the inside. So this is the critical thing, is the potassium. On the outside, the critical thing is that there's a higher ratio of sodium ions. There's also a higher ratio of chloride ions, which is interesting because the chloride ions are negative and the sodium is positive. But there is so much positive sodium that it will still keep a higher positive charge on the outside, and this will be a lower charge, which we translate as negative, although the thing that's really determining it is still a positively charged ion. It's just that there's less of them on the outside than on the inside. Now, if we look at this picture, I've got leak gates right here, okay, and here. Notice that they have different things going through them. So this one allows sodium through, and this one allows potassium through. So potassium is going to follow its gradient from high inside to outside, okay? Whereas sodium is going to follow its gradient from high outside to inside, okay? Now, if we let that go on, basically then we would end up with an even amount of potassium on both sides and an even amount of sodium in both sides, and that would totally screw up your ability to have a resting membrane potential. So one thing you're going to do is you're going to throw in the sodium-potassium solute pump. So this thing right here. This becomes really important for helping to maintain that ratio. What it's going to be doing is it's going to be chucking things in the opposite directions, so that it basically is going to push the sodium out and pull the potassium back in order to maintain the 3:2 ratio of sodium to potassium, okay? And this basically keeps more potassium inside and more sodium outside as well, okay? All right. Let's look at the next picture. All right, here we're basically looking at the same thing. In this case, we've got -70, so we're dealing with a neuron, okay? And they're basically making the argument with the bigger Na, saying I've got more on the outside, so 150 to 15, and then with potassium, it's 5 to 150. So it's basically creating a negative charge here and a positive charge there. Okay, what I like about this picture though is what they've done with these arrows. So you see with potassium they've made a really fat arrow, and with sodium they've made a very narrow arrow. And what these are representing are those passive leak gates, okay? So potassium is 150 on the inside, on the oh, what did I just do? Yeah, I reversed it. Potassium is 5 on the outside and 150 on the inside. So actually, let me write this differently. So I've got, all right, which means I've got a really steep gradient. It's going to flow faster because the gradient is more intense, okay? Whereas over here with sodium, I have 150 here, and I've got 15 here. Sodium's gradient is less steep, so it does not flow as fast. So not only do I have these leak gates, but they have different speeds of movement, which basically means we can say they have different permeabilities. Okay, so the plasma membrane, so the PM, the plasma membrane, is more permeable to potassium than to sodium. Now remember, this is all through those passive gates, or those passive channels. Okay? So they're going to be following this. Now, again, if you just let this go, it would eventually be equal on either side. So we're going to throw in a very important structure, the sodium-potassium solute pump. And what that's going to do is it's going to use ATP energy to take these things and move them in different directions. So it's going to take, so the sodiums are going in this way, so we're going to use the solute pump to take the sodiums back out that way. Okay. All right, let's change the color. So the sodium wants to go this way, so I'm going to use energy to take it back out that way. Okay. And then potassium wants to go in that direction, so I'm going to use the solute pump to do that, to take it back into the cell, so that I can maintain that ratio of 3 to 2. Okay? All right, let's look at the next picture. And actually, this is the same picture, but I've got different notes on here where I'm basically just repeating what I've said on the previous. High permeability to potassium. Potassium is more concentrated inside the cell than outside. Plasma membrane has a lower permeability to sodium. Okay? One thing to remember is because potassium has a stronger or steeper gradient, it's actually more important for maintaining that negative 70 millivolts or negative 90 millivolts. So it's more important for maintaining this resting membrane potential. Okay? And then right here, I've just got the numbers right there for resting membranes potentials. All right, let's look at the next slide. All right, in this picture, I'm taking the diagram from the previous page, which I like because they use arrow width to symbolize permeability, okay, through those passive gates. What I like about this picture is instead of arrows, they've got the actual gates, okay? So orange is sodium, blue is potassium. I don't know why I did that, okay? The other thing I really like about this picture is that they're showing lots and lots of sodiums out here, not a lot of blue potassiums, but on the inside, lots and lots of blue potassium and only a few of the little orange sodium molecules. So you get this really good feel for how much potassium is here and how out here on the outside there's way more sodium, okay? And then you can see that this gate is narrower, all right? And partially that's because this ratio of sodium, so here they're showing sodium 145 to 12 because it's a different structure, but anyway, don't worry about it. It has a narrow gate, but over here, because the ratio is 150 to 4, the gate is wider because there's more pressure on it, like a bigger hose, and so more potassium can go out at any one time, okay? Now, this is, again, all passive right here, so we do need to use that sodium-potassium ion pump in order to keep fixing this ratio that's going on. So this is following concentration gradients, and if you just let this thing go, eventually you would have an equal number of sodium and potassium on both the inside and the outside. So it's constantly using the sodium-potassium pump to fix that ratio. All right, let's look at the next picture. All right, so here's the picture we've been looking at on the previous slides, okay? And now we're going to look at this particular picture right here. And these first gates are these passive gates. And then over here is the sodium potassium ion pump. Okay, now here they're symbolizing the greater permeability of potassium by showing 2 channels instead of 1. Okay, so I want you to think about why there's more potassium inside and sodium outside. Okay, this is going to be this active process controlled by this. Okay, so the sodium potassium pump is going to be drawing the potassiums back in and taking the sodiums out in order to maintain that ratio. But you always have this passive process going on. More potassium can slip out. Okay, so you've got this constant dance between passive diffusion and active pumping of things in order to maintain that ratio. Okay, all right, let's look at the next slide. All right, here we're basically kind of just following up about some and want to talk about some things that are going to happen. Okay, so if more potassium, just change color, if more potassium, and remember potassium is a positive ion, leaves the cell along its concentration gradient, so that's right here, it's leaving the cell through its concentration gradient, what kind of charge does that leave inside the cell? So if more potassium leaves, you're taking more charged ions out. So that's going to leave fewer positive charged ions. So overall, the charge inside is going to become more negative. Okay? So what 2 things are maintaining this higher concentration of ions outside the cell? So you've got this sodium potassium solute pump. All right, that is doing it. Okay, but also you've got the passive leak of specifically positively charged potassium ions. So those are constantly leaving. So that is also going to mean that overall there are more positive ions outside the cell. So more positive outside the cell, okay, because technically potassium leaks out of the cell faster than sodium leaks into the cell. Okay? All right. So let's follow up on one more slide and just do some follow-up notes. All right. So we're going to change membrane potential, okay? How can we change it? How? Okay? So first, you've got alterations in the permeability of that membrane to certain ions, okay? Potassium is more permeable, sodium is less permeable. That affects membrane potential, okay? And then we're looking at the concentration of charged ions on either side of the plasma membrane. That's the second thing that's really affecting it. Okay, so these are the 2 main players. Now, in the next couple of slideshows or video lectures, we're going to start talking about local potentials versus action potentials. Mostly so far with the muscular system and the introduction of this chapter of neurophysiology, we've focused on action potentials. But local potentials are always happening as well, and they are not self-propagating. And we'll talk about that more in a little bit. Definitions. You need to know what these things mean. So depolarization, repolarization, and hyperpolarization. Okay? This is when you're going to change the permeability, you're going to change the charge, change the polarity. All of these are changing the polarity. Okay? Here you're making it less negative. With repolarization, the goal is to get it back to resting membrane potential, which basically means you are making things more negative. okay? Hyperpolarization is when you make it too negative, okay? That resting membrane. So let's actually review this. Resting membrane potential is when you are talking about the polarity of the plasma membrane. And basically this is going to be about the ratio of ions on either side. And we measure this in the voltage or charge difference between the two sides. All right, and the two numbers that we've got you memorizing are negative 90 millivolts for muscles and negative 70 millivolts for neurons. Okay? Now, graded is a synonym for local. Well, it's not really. Actually, let me erase that. It's sloppy anyway. So graded is talking about degree. Local is talking about location. Okay? And we can call the same thing either a graded potential or a local potential, and it's basically describing the same thing. With these, okay, they can be depolarizing a membrane, or they can be hyperpolarizing a membrane with local potentials and graded potentials. That's an important thing to remember, okay? So it can be one of those two things. The other thing to remember is that these tend to be short distance, all right, which is why we call them local, all right, and short-lived. They fizzle out fast, okay? In contrast, action potentials, they go a long distance. They're going to live as long as there is a path, all right? Because they are self-propagating, okay? Also, they open voltage gates, which means once they start, they do not stop, all right? So they are all or nothing, all right? They go to the end, all right? Graded potentials, by the way, are primarily opened by chemical gates, or stimulated, not opened, but started by chemical gates or mechanical gates. Okay? All right, we're going to stop the recording there, and then we'll move on to the next video lecture.